My name is Da Liu, affiliate 1 School of Life Sciences, Beijing University of Chinese Medicine. I introduced the paper submitted to iMeta. The title is Decoding the Role of Immune T Cells, a New Territory for Improvement of Metabolic-associated Fatty Liver Disease. Firstly, metabolic-associated fatty liver disease MAFLD is a new emerging concept and associated with metabolic dysfunction, generally replacing the name of non-alcoholic fatty liver disease NAFLD due to heterogeneous liver condition and inaccuracies in definitions. More shockingly, the number of MAFLD is continuously increasing and may expand to 100.9 million cases in 2030, which affects up to 25% of the global population and plays a growing impact on economic development, public health, and social determinants. Nevertheless, MAFLD still encounters the current dilemma due to unclear pathogenesis and the lack of FDA-approved drugs in clinical treatment. Currently, in addition to the common causative factors like insulin resistance, oxidative stress, and lipotoxicity, the role of immune cells, especially T cells, play in MAFLD is increasingly being emphasized by global scholars. The immune system is a collection of cells and molecules that mediate immune responses such as allergy, tumor immunity, and autoimmunity, which can be roughly divided into innate immunity and adaptive immunity. We analyzed the physiological characteristics and function of different T cells, including Th1 cell, Th2 cell, Th20 cell, Th17 cell, tract cell, CD8 positive T cell, gamma delta T cell, and T cell and mate cell. The clinical and animal studies were observed the accumulation of inflammatory immune cells like Th1 cells, M1 macrophages, Th17 cells, CD8 T cells, and gamma delta T cells in the liver or adipose tissues, while the decrease of the quantity and dynamism of anti-inflammatory cells such as Th2 cells, tract cells, and T cells. These observations imply that the destruction of immune homeostasis may cause hepatic inflammatory responses, liver fibrosis, or further hepatocarcinogenesis through mediating inflammatory cytokines and chemokines-related signaling pathways. In addition, CD8 T cells and T cells acted as a double-edged sword that could either exacerbate steatohepatitis or improve MAFLD in different in vivo models. Secondly, some gene knockout experimental animals were more likely to suffer from MAFLD and even have symptoms of severe metabolic disorders. Furthermore, it has been found that both the imbalance of the intestinal immune system and the homeostasis of intestinal microbes can also cause the imbalance of T cells and the dysfunction of liver metabolism. We comprehensively analyzed the interaction between MAFLD and T cells, as well as the relationship between hepato-intestinal immune crosswise and intestinal flora homeostasis, and pointed out several T cell-based treatments, including but not limited to adoptive T cell transfer, fecal flora transplantation, and drug therapy, such as the use of cytokines or chemokine inhibitors or neutralizer. It is especially emphasized that natural products and Chinese herbal compounds can not only regulate T cells, but also regulate the balance of bacteria to treat MAFLD and restore the healthy state of liver. In summary, we first reviewed the pathogenesis of MAFLD and the function of different T cells. Secondly, we deeply summarized and analyzed how different T cells interact with each other to participate in development of MAFLD. This current study not only points out the importance of immunomodulatory function of T cells in the process of MAFLD, but also provide new ideas and breakthroughs for the discovery and development of further drug trials and mechanistic studies.